Okay, in order to move on, we need to review a concept you learned in chemistry, oxidation and reduction. Now relax, we aren't going to make you write and balance equations or do some stoichiometry, so take a deep breath. What you have to be able to do is identify the oxidizing and reducing agents as well as the substances being oxidized or reduced. The acronym oil rig is the easiest way to remember this process. Okay, so get this diagram down in your notes and let's write some statements that explain it. First, whenever a substance loses electrons, it's said to have been oxidized. So when it loses electrons, it's said to have been oxidized. Okay, so because A, in this case, loses some electrons, A is being oxidized. Now, the substance responsible for the loss of these electrons is called the oxidizing agent. In this case, it was substance B. So substance B was responsible for stripping these two electrons from this reducing agent and oxidizing A. That's why this blue line is pointing down here, because B stripped these electrons now and A became oxidized. Now, whenever a substance gains electrons, it's said to have been reduced. So in your diagram, you see that B has gained now the two electrons. It's gained two electrons, therefore it's reduced. Now, the substance responsible for giving these electrons is called the reducing agent. So A had the two, it now got stripped by B, and B now has the electrons, right? So this is the reducing agent, and B is the substance that's been reduced. So again, A is oxidized because it's lost its two electrons. B is responsible for that, so it's the oxidizing agent. B's reduced because it's gained the two electrons and it got them from A, so A is the reducing agent. I know that seems a little bit complicated, but what you need to remember in its basic form is what's being oxidized and what's being reduced, and we'll show that in this photosynthesis process. Okay, remember, there are three organisms that photosynthesize: plants, cyanobacteria, and blue-green algae. Now, we're going to separate them into cyclic and non-cyclic photophosphorylation. Don't panic. You're going to understand it. You don't right now, but you will soon. So, let's start with plants and cyanobacteria. Plants and cyanobacteria utilize two photosystems. We saw that in the previous diagram. And they work together to produce both energy and reducing power. Now we're going to devise a series of steps that describe each of these processes. So be ready to write these steps underneath the diagram you wrote with photosystem 1 and photosystem 2 in it. Okay, here we go. Write in your notes that when a photon of light from the sun or other sources like the regular lights in the room strikes the reaction center of photosystem 2, it excites an electron. Okay, so here's our electron now, our photon of light exciting an electron. Now, in your notes, write that the reaction center in photosystem 2 is also referred to as P680. Now, the reason it's referred to as P680 is that's the wavelength of light that PS2, photosystem 2's reaction center, is sensitive to, 680 nanometers. So write that down now. Now, remember from chemistry now that exciting an electron means temporarily bumping it up to the next energy level. So when we see this electron now go this way, it's now being bumped up to another energy level. It's called an excited electron. So here now is our excited reaction center. Okay? So our electron now started in a lower energy state, at ground state, and then that photon of light, boom, excited it so that it now is in an excited reaction center. Now, at the same time, two water molecules bind to an enzyme, you see it right here, called the water splitting enzyme, connected to the photosystem 2 reaction center. I know, right? All the names they could have chosen, they picked the water splitting enzyme. So here you see it right here, the water splitting enzyme. So two water molecules get split. So that's done with step 1. So again, in step 1, photon of light strikes the reaction center. So here's our photon of light strikes the reaction center, excites the electron. And then now two water molecules get split by an enzyme called the water splitting enzyme that's connected to the photosystem 2 center. All right, on to step 2. Okay, now on to step 2. In step 2, this water splitting enzyme splits water and strips it now of some of its electrons, which are used to replace the original excited electrons in photosystem 2, while at the same time oxygen is then given off into the atmosphere. Yes, the breathable kind that keeps us alive. It comes from water. That question is almost always on the AP exam somewhere. So where does breathable oxygen come from? Well, you can see right here now that the electrons are used, all right, from the stripped water molecule, and that oxygen, you see the oxygen produced right here, is given off into the atmosphere. So the breathable oxygen comes from the splitting now of this water molecule. In fact, water becomes an extremely important part of this photosynthetic process. So the electrons are going to be used to replace the electrons lost in photosystem 1. The oxygen is released into the atmosphere, and we're going to see what happens to these hydrogen protons right here very soon. Now, we're going to call the electron now. You see this electron passing down this thing? This is called an electron transport chain. So we're going to call it an electron transport chain, and then we're going to watch this electron move down it very quickly. So in your notes, write the words electron transport chain. All right, now we run into our first enzyme in this process called Plastoquinone. All right, so we're going to see our electron now make its first step down this electron transport chain. So Plastoquinone is an enzyme, right? It's an electron acceptor. So we're ready to write step 3. In step 3, Plastoquinone accepts the original electrons from the pigments excited by the light in photosystem 2. And it passes these excited electrons to a proton pump embedded in the membrane called the B6F complex. So let's look at this. You should be writing these steps in your notes. So boom, electron Plastoquinone to the B6F complex. Now remember, because it gains electrons, it's being what? Think of oil rig. Yes, it's being reduced. Now's a good time to review some more basic chemistry. Remember when you studied acids? When hydrogen loses an electron, it becomes a positively charged ion, right? The hydrogen ion. Well, in chemistry, we also refer to this as a proton. Can you remember why? Yes, because the element hydrogen has no proton. So when it loses its only electron, or has no neutron. So when it loses its only electron, all that's left is a proton. So we're going to refer to hydrogen ions. And let's go back now. And here's our hydrogen ions. We know they're ions because of this plus. Why is it plus? Because we stripped the electrons from them. Okay, we stripped them the electrons. So they're hydrogen ions. We're going to refer to them as protons because all that's left is a proton. Right? Hydrogen has one electron, one proton, and no neutrons. So we're going to use those interchangeably. Okay, now on to step 4. Now, arrival of these electrons into the B6F complex causes the B6F complex to pump protons, otherwise known as what? Hydrogen ions, from the stroma into the thylakoid space, which now sets up what's called a proton gradient across the membrane. So let's take a look at what that looks like. Okay, so here's another visualization now. We see now the stroma here. We see the thylakoid space. And now we see our photon striking photosystem 2. All right? And here's its electron now, Plastoquinone. Here's our water splitting enzyme, now replacing that electron that was lost. And now we see the hydrogen atoms now scattered. There's 4 of them because how many water molecules split? 2. All right? And oxygen then is going to get released where? Into the atmosphere. So here's our electron transport chain. Now remember, we're working up to step 4. So our electron now gets passed to Plastoquinone down this electron transport chain via the B6F complex. Now, let's write step 4. Now, arrival of these excited electrons causes the B6F complex to pump protons, otherwise known as hydrogen ions. Here they are. All right, they were outside, see? They were outside. Now they're going to pump, the B6F is going to pump protons or hydrogen ions into the thylakoid space. So in step 4, B6F pumps hydrogen ions into that thylakoid space. Now, what this creates now, or it's going to create, is a proton gradient. Remember what a gradient is from osmosis and diffusion. It's an area where high concentration meets low. So as this thing keeps pumping hydrogen ions in there, and here we go, we're going to see this fill up now down here with a hydrogen ion. Now that's important now. These are important molecules. All right, let's move on to step 5. Now, in step 5, we're going to see how important these protons are. So now we're pumping these protons in, and in step 5, as protons pass through the membrane, so you can see now they're passing into the membrane here. And now we're going to use them and their energy in another way. Remember, the idea here is to make ATP, to make energy. Okay, that's kind of what we're looking at with this electron transport chain. So now let's look at the role of this proton. So this proton then is pumped back through, and now its energy is used to reunite this phosphate and this ADP, adenosine diphosphate. And remember, if we add a phosphate to ADP, what do we have left? ATP, that's correct. So let's look at that process again. Now, boom, there's our ATP. Now, we call this process photophosphorylation. Photophosphorylation. P-H-O-T-O-P-H-O-S. P-H-O-R-Y-L-A-T-I-O-N. Phosphorylation simply means to add a phosphate. So ADP is phosphorylated using this hydrogen ion, right, to provide the energy into ATP in step 5. Now, of course, any reaction needs an enzyme. Why? To speed it up. So here you see ATP synthase. What ATP synthase does is lower the amount of activation energy it takes to phosphorylize this ADP molecule. So make sure you write that in your notes. So ATP synthase facilitates this phosphorylation. In other words, it reduces the energy that's necessary to phosphorylate ADP into ATP. Okay, so let's back the bus up here. So here we have now our excited electrons moving down the electron transport chain, being replaced now in photosystem 2 by the electron stripped from water. And here comes the electron, use now its energy to pump these protons in here so they can be used to phosphorylate ADP into ATP using the ATP synthase molecule, which is an enzyme. It's a protein. Now, let's go to step 6. Okay, so now we've got photosystem 2, and now we have photosystem 1. So we've just seen now the electron transport chain action in photosystem 2. Now remember, the excited electrons now are moving down this electron transport chain and are going to replace the electrons in photosystem 1. And the electrons that were lost in photosystem 2 are replaced by the water molecule that was stripped of its electrons in this water splitting enzyme right here. Oxygen was released into the atmosphere. The electrons replaced these electrons, and the hydrogen ions were used to phosphorylize ADP into ATP. Now let's look at what happens in photosystem 1. So we're on step 6. When photosystem 1 absorbs its now photon of light, its own reaction center passes excited electrons not to Plastoquinone. That wouldn't make sense because it's not in photosystem 2. It's a different now acceptor called ferredoxin. So it passes its electron now to ferredoxin. So make sure you write that in your notes. Now then, the enzyme NADP reductase, here's the enzyme NADP reductase, transfers electrons to NADP, here we go, to form NADPH. There's our NADPH. So while ATP now was made in this original step, NADPH is made in this second step. So the outcome of the light reactions are two molecules, ATP and NADPH. Now let's write step 7. In step 7, the electrons lost from photosystem 1 are replaced, here we go, by the electrons now from photosystem 2 that were stripped of the water molecule by splitting it. Now, this is an important addition to this step 7. Because the original excited electrons in photosystem 2 do not return to photosystem 2, this process is referred to as non-cyclic photophosphorylation. Non-cyclic photophosphorylation. So make sure you get that in your notes. All right, let's look at this whole process again. So here's this whole process again. So our light strikes our photosystem 2, water split. All right, the original electrons now travel down the electron transport chain. Oxygen released into the atmosphere. Water replaces those electrons. All right, BF6 complex pumps in the hydrogen ions. And then ATP synthase uses those hydrogens to phosphorylize ADP into ATP. And in photosystem 2, light strikes, ferredoxin transfers the electrons to NADP reductase, which now uses that energy to reunite NADP and H to form NADPH. And the electrons from photosystem 2 are replacing the ones that are lost here. All right, that's 7 steps in non-cyclic photophosphorylation.